Thank you for the introduction and good afternoon. As mentioned, my name is Anders Ljungqvist. I'm the CEO of the Scandinavian, Swedish-Danish biotech company called RhoVac. RhoVac is working with immuno-oncology, and we are focused on treatment of metastatic cancer. What we mean more specifically on the metastatic cancer is that we are developing a cancer vaccine. A cancer vaccine that is focusing on T-cell activation and a vaccine that is focusing on metastatic cells. We do that by targeting the protein RhoC, and RhoC is known to be overexpressed in all metastatic cancer cells, regardless of from where they originate. So the product has a very broad potential from an indication point of view. And in addition, any therapeutic cancer vaccine has a very good safety profile and is a targeted therapy, meaning that it's suitable for combination therapy. Now the people, or at least some of the people behind this vaccine, if we begin at the bottom, are the two professors from the Copenhagen University, Dr. Andersen and Dr. Per Thor Straten. They are the inventors, and they are still the scientific advisors of the company. As you can see from the publication, they have a lot of scientific experience, but they also have made a lot of commercially interesting things, and currently 3 of their inventions are in phase 2 clinical trials in different companies around the world. Henrik, our CFO, has a long experience, latest with Santaris Pharma, which was sold to Roche in 2014. And myself, I have more than 30 years experience in life science, 25 years at Leo Pharma, and 13 years as an independent consulting, doing coordination and planning of drug development. In addition, we have a very experienced board that will support us very much in our development. What I'll do now is just jump into the status of our ongoing clinical trial. It's a first-in-man trial, phase 1/2 trial, performed in prostate cancer patients. Enrolled into the study are 22 patients. It is it's a first-in-man trial, so it's a safety trial, primarily looking at safety and tolerability of the vaccine. Secondarily, we are looking at the immune response that we are raising with this therapy. And thirdly, we have some explorative objectives looking at any link between the PSA values and the immune response that we find. And we are evaluating endpoint like progression-free survival in this, and this primarily for planning for the phase 2b study that we hope to start sometime next year. Status is currently that we recruited the patients, the 22 patients, and that was finalized in July, early July this year, and it took 3 months, which is very fast. Currently, all patients have received 8 vaccinations out of the total 11 vaccinations that they will receive during the treatment period. And 1 patient have received 10. That is a correction because this morning he received his 11th and final vaccination. All patients will be finalizing their treatment period by mid-February 2018. Following that, we will do the immunological analysis, and we plan to report the study in second quarter of 2018. So, why do we believe that RhoC is a different and a very attractive cancer antigen? First of all, it's a self-antigen. It's silenced in normal cells, and there's no interaction with the immune system. In metastatic cells, the RhoC is overexpressed and is regarded by the immune system as foreign. And if we teach the cells to do so, they will attack and eliminate metastatic cancer cells. When we are developing new concepts, or rather the professors in the university, they are always looking for antigens that play a crucial functional role in the cancer cells. So it's survival or progression. And in the case with RhoC, the functional role is the motility of these cancer cells. It has been shown that if you reduce the expression of RhoC, the motility will be reduced, and in doing so, the number of metastatic cells will be reduced. As mentioned, RhoC is overexpressed in practically all metastasizing cells, regardless of the origin of the metastasis. And in addition, it has been found that RhoC is also overexpressed in cancer stem cells. And a number of publications are indicating that RhoC should actually be further developed and further investigated as a target for treatment of cancer stem cells. We at RhoVac does not have big experience in that, so for that reason we have entered into a collaboration with the university in Lund, and there is a group there that work with prostate cancer stem cells for about 10-15 years, and they are currently evaluating if the overexpression is still the case in prostate cancer stem cell and looking at potential effect on using our vaccine. It's also known that mutation is very rare in the protein RhoC in cancer cells, and this is a clear indication that the effect is related to the overexpression, not to other activities of the protein. This means that immune escape is probably not a problem when using RhoC as a target. In addition, finally, National Cancer Institutes have listed the antigen RhoC as a prioritized antigen in their publication of 2009. So, when developing a cancer vaccine, it's always difficult to use animal models, and currently there are no validated animal models. So what we have done is to use an ex vivo model, and this is the proof of principle that the authorities have also accepted as proof of principle for going forward into clinical trials. So first of all, it's a three-step approach. We investigate whether our antigen can bind to the HLA molecule, and this is the case. And we're measuring that with the binding assay, which is a standard measurement. Secondly, we do investigate if we can raise the spontaneous immune system against RhoC, and this is measured with ELISpot, interferon gamma ELISpot, and this is shown to be the case. And then thirdly, what we did was to take the T-cell clone we have done in the second step and see if this T-cell clone can eliminate human cancer cell lines. The T-cell clone used is from a melanoma patient, and if you look at the green light, you will see that the increasing ratio between the T-cell clone and the cancer cell, you will see an increased elimination of the cancer cell. So it does work. But more interestingly, I think it is here to see that we use the same T-cell clone from the melanoma patient to show that we also could eliminate cancer cell lines, breast cancer cell lines, and colon cancer cell lines. Again, supporting the concept that the RhoC is the overexpression in metastatic cancer cells regardless of the origin of the cancer. So, looking back in history, we have seen a number of failures or vaccines not functioning up to expectation. And we believe with the RhoC that we have some differences. First of all, it's a cancer vaccine where we are targeting early stage of the metastasis. We are not targeting the mother tumor or a heavy tumor load. And we believe that this is one of the reasons why earlier vaccines have had problems with success. Secondly, the immune resistance is not that likely because The RhoVac is not developed until the cancer cell starts to metastasize. They are not present in the mother tumor. And that means that they are available or they are overexpressed at the time of diagnosis. So for that reason, immune resistance is not that likely when you work with RhoVac as a target. And finally, in the old days, you very often see that it was short peptides, 8 to 10 amino acids that were used. And these do activate CD8 T cells, but they do not activate CD4 cells. To activate CD4 cells, you will have to have an amino acid sequence of at least 17 amino acids. Ours is on 20, and we do activate CD4 and CD8 T cells. This means that we provide a better environment for the immune system. Now, this leads that we are looking for an intervention point right after a finalization of treatment of the primary tumor. And the concept we are working with is treat the primary tumor with conventional treatment, being chemotherapy, being surgery, or radiation, and then control metastasis with a cancer vaccine that is targeting RhoVac. You can then discuss whether we are going for monotherapy or combination therapy, because I mentioned to you that it's very suited for combination therapy, checkpoint inhibitors, obviously. In our case, we would like to start to go for monotherapy and doing this in micrometastasis, early, very, very early in the stage of metastasis. The labeling that we are trying to develop here is a labeling that we call adjuvant or maintenance treatment against metastasis following completion of treatment of primary tumor. And this labeling that we want to develop is a labeling that is true for any metastasizing cancer indication. What we are doing in the phase 2b trial, our plans for that is to have a trial where we have some endpoints that could be disease-free survival, metastasis-free survival in combination with a biological endpoint. It will be a study taking about 3 years, 1 year for recruitment, and 24 months to treat and follow up on the study. And we define a patient group which is existent, where it's expected that 50% will have relapse within 1 to 2 years. In addition, we would like to conduct some explorative studies, but that will be without placebo-controlled, and it will be historical endpoints, historical comparison. So we are finalizing now the initial part of our development, and it will be finalized mid-2018. And we are keeping the timeline, and we are keeping and being on budget. So what we will be looking for is funding for the period of 2018 to 2021, where we will be doing the regulatory interaction. We have already had a very good and positive SME briefing meeting with EMA, and we are going for scientific advice with submission in beginning mid-February. After completion of this study, that is a relative comprehensive phase 2b study, we are trying to do that because if the data are good, we potentially could send in a submission for an accelerated or conditional approval, meaning that we could reach the market much faster than we would if we had to complete the full phase 3 clinical trial. And in parallel, we are conducting and continuing the research collaboration. We have the University in Tübingen, experts in immune monitoring, with Center for Cancer Immune Therapy, that's a professor that we're working with, and Lund University with cancer stem cells. So the business opportunity is that we are a public traded company in Sweden. I think we have a unique immune oncology product, and with potential of very, very broad treatment indications. We have a product that is currently in phase 1-2. It's on track, it's on budget. We have a market potential with more than 10 million new cases of cancers every year. Patent coverage is good in Australia, in US, Japan, in Europe. Canada is still pending. In this development, there are multiple value inflection points. In the business approach that we have currently, we are looking for license or sell all or part of the operations after completion of the clinical phase, which means mid next year. But we are open for partners that would work with us in conducting the phase 2b clinical trials. And currently we are discussing with potential investors and potential partners. With this, thank you very much for listening to my presentation.